Good evening, this is Mr. Wintermelon. So, in the previous few chapters, we have learned a lot about your chromosome as well as your gene. But what we have not explored is the basic molecules which make up your chromosome, which is DNA. So, in this chapter of molecular genetic, we are going to learn more about the structure of your DNA as well as what mad scientist experiment we can do with your DNA. In the previous chapter, we have mentioned that your DNA codes for protein. But how exactly does your DNA do it? We will answer this in this chapter. So just a brief overview. In this video, we will learn about the detailed structure of DNA. Then, as mentioned, we will answer how does DNA codes for protein. But it is just a very watered down version for your O-level. The exact detail is very complicated. So I may make a bonus video for it, but the exact detail of how your DNA becomes protein or codes for protein will not be totally covered in this video, because for your O-level syllabus, you just need to know 5 words when it comes to DNA coding into protein. In the next video, we will learn about genetic engineering. What fanciful experiment we can do with our DNA. So by the end of this lesson, you should be able to outline the relationship between DNA, genes, and chromosome, as well as stating that a gene is a sequence of nucleotides as part of a DNA molecules. The first 2 learning objectives, we actually touched on that before, so not a big deal. The new thing that you will learn is the more detailed structure of DNA, as well as the rule of complementary base pairing. Then lastly, you will learn how DNA carries the genetic code which is used to synthesize specific polypeptides. So let us just do a quick recap of the relationship between DNA, genes, and chromosome. DNA coil around the protein histone to form chromatin. During cell division, chromatin will replicate to form sister chromatids, which will then condense into your chromosome. So this is the relationship between DNA and chromosome. Gene, on the other hand, is a sequence of nucleotide in the chromosome which codes for protein. So you will see a new word which I use down here, nucleotides. We will learn what is nucleotides in this lesson. Now, one question that we have not answered is, since all the DNA in your cell is the same, but how come all our cell or different cells in different organs behave differently? That is because, depending on the cell, certain genes are switched on and off. That means in your stomach, your pepsin gene will be switched on, but in your small intestine, your pepsin gene will be switched off, and instead your peptidase gene will be switched on. So depending on the location of the cell, certain gene will be switched on and certain gene will be switched off. So let's go back to the important question of the new word, nucleotides. What is it? Nucleotides is actually the basic unit of your DNA. So the basic unit of DNA is known as nucleotides. Most of the time, it will be represented in this form. So you should be able to see that there are 3 main parts. Your nucleotides will consist of a phosphate group, a 5-carbon sugar, as well as a base. Why do I put a question mark down there? Because there are 4 possible bases which the nucleotides will have. First, we have our adenine, thymine, cytosine, as well as guanine. So those are the possible bases which your nucleotides can have. Please remember the spelling is not too difficult because they sound like how they are spelled. Adenine, thymine, cytosine, and guanine. Repeat after me. Adenine, thymine, cytosine, and guanine. So repeat a few times and you should get the hang of it. So this is the monomer of your DNA. How about the whole structure of your DNA? So just like your protein, which consists of many amino acids, your DNA will also consist of many nucleotides. And those nucleotides covalently bond. You see the covalent bond. Let me repeat for the third time. To form a strand of polynucleotides. A very creative name. No one would have thought that many nucleotides will be called polynucleotides. Do you have to know that it is covalent bond? Not really. But since you probably have learned covalent bond in chemistry, why not use it? So many nucleotides will covalently bond to form a strand of polynucleotides. Something which is not in your syllabus is the name which we give to the sugar and phosphate group, which kind of looks like a backbone. So the creative name given to the sugar and phosphate group that looks like a backbone is the sugar phosphate backbone of the DNA. So the sugar as well as the phosphate group, or the yellow color line of the DNA molecules that you see down there, is known as the sugar phosphate backbone, just for your information. But of course, DNA is not just one single strand of polynucleotides. You can see from the 3D model, it is actually double-stranded. So the other strand of DNA will bond to this original strand in this manner. As you can see, it is like upside down. Something like your 6 and your 9, rather than a 6 and a 6. DNA doesn't bond that way. DNA bond in the form of 6 and 9. But of course, the name is not DNA bond in a 6 and 9 manner. We call this manner antiparallel. So two strands of antiparallel polynucleotides will bond to form your DNA. So the bond between the two strands of antiparallel polynucleotides is hydrogen bond. Again, hydrogen bond is FYI, more for your info. And these two strands, they will twist and form a double helix, which you can see down here. So this is the structure of DNA. It is made up of two strands of antiparallel polynucleotides, which twist to form a double helix. Now, I want you to take a closer look at the base pairing, or how the bases are paired up. Does adenine bond with another adenine, or can adenine bond with any other bases, like adenine bond with thymine, adenine bond with cytosine, or adenine bond with guanine? Same thing for the other bases, like thymine. Does it form bond with another thymine or with other bases? Pause the video and see whether you can see any pattern or not. So if you examine the base pairing carefully, you should be able to see that adenine actually only form a bond with your thymine, while cytosine only form a bond with guanine. why there are two dotted lines for adenine and thymine is there are two hydrogen bonds between adenine and thymine, while there are three hydrogen bonds between cytosine and guanine. So you have to remember this complementary base pairing. A bond with T, C bond with G. But of course, you need to know the spelling. It is not difficult to remember this base pairing. How I go about remembering this is I look at the letter C and the letter G. They are like kind of similar. So C will bond with G. And of course, A must bond with T. And that's about it. So to summarize the structure of DNA in words, DNA is a molecule composed of two anti-parallel polynucleotide chains that coil around each other, forming a double helix. Those polynucleotides is covalently bonded by many single nucleotides. Now, I must emphasize on the words that are in red. Those are the marking point of O-level. So as you can see, the covalently bonded is not part of the marking point because this is biology, not chemistry. So just take note of the key point, which is anti-parallel polynucleotides chain as well as double helix. A nucleotide consists of a phosphate group, sugar, and one of the four bases, adenine, thymine, cytosine, or guanine. So you will be awarded mark by stating phosphate group and sugar. The four bases you will also be given mark, but it is only when you mention about the complementary base pairing. Bases of the two separate polynucleotide strands are bonded by hydrogen bonds, with adenine complementary to thymine and cytosine is complementary to guanine. So those are the marking points if they ask you about the structure of a DNA molecules. So now that we have learned about the structure of DNA, let us proceed to learn how DNA codes for your polypeptides or your protein. But first, let us talk about some codes which you might be familiar with, which is the computer code. Computer actually use something known as binary codes. Binary, BI, that means 2. So for all computer, if you do not know this, they are coded using 0 and 1. So I have a list of the binary code down here. You can see that 0011 0000 codes for 0. Then 0101 0100 codes for T, and so on. So your coding system of binary code is something like how your DNA codes for your protein. So this one is more for your information. Let us compare binary code and DNA. For binary codes, it uses two numbers, 0 and 1. DNA uses 4 nucleotides, A, T, C, and G. Binary codes code for characters inside the computer, while DNA codes for amino acid. Binary codes uses 8 numbers to code for 1 character as seen in the previous slide. In the case of DNA, 3 nucleotides is known as 1 codon, and 1 codon will code for 1 amino acid. So if you are curious about how this codon works, I actually have this diagram down here. Just take note, your thymine is replaced by U, T is replaced by U. If you are curious to find out why, you can read up more by yourself. But how your DNA codes for amino acid is in this way. For example, if the first letter in the codon is A, second is U, and last letter is G, it will code for the amino acid methionine. If the first amino acid is cytosine, second is cytosine, and third is cytosine, it will code for proline. If first and second are cytosine and the last is guanine, it will also code for proline. So this chart will tell you, depending on the first, second, and third codon, which amino acid the codon will code for. Some codon will code for the same amino acid. So if the first base is U, second base is A, and the third base is A, for example, it will actually not code for any amino acid. That means no more amino acid will be added to the polypeptide chain. So again, if you do not understand this, don't worry, it is totally not in your syllabus. It is here just in case you are wondering how does the combination leads to the 20 amino acid. So an analogy for how DNA codes for protein is DNA is like an instructional manual to make yourself. So instructional manual needs words, and your DNA is like your simplified Chinese. So the key thing to note is the words that are in red. 5 words, which I mentioned at the start that you have to know. So DNA is something like your simplified Chinese. Then DNA will undergo transcription into mRNA, which is like your simplified Chinese get rewritten as traditional Chinese. It is not a translation because both of them are still the same language, the Chinese language. While DNA and mRNA, they have the NA down there. NA stands for nucleic acid. Both of them are still nucleic acid. So mRNA, the M down there stands for messenger RNA. And the function is it serves as a messenger to be translated into protein or your polypeptides. Why is it a translation? Is because when you translate Chinese into English, it is a translation. So from simplified Chinese, we transcribe into traditional Chinese. Why is mRNA traditional Chinese? Is because RNA is more ancient than DNA. That means RNA appeared first before DNA. That's why the analogy for RNA is traditional Chinese. Then from the Chinese language, we translate to another language, English, which is your protein, because protein is not nucleic acid. That's why from mRNA, it is translated into protein. And again, I like to emphasize that you just need to know 5 words. DNA undergo transcription into mRNA, and mRNA undergo translation into protein. And we just have to know a little bit more about translation, but plot twist, you kind of already know translation. We have actually mentioned it in the first chapter of cells, just that we didn't use the word translation. So all you have to know is this. DNA is transcribed into mRNA. This is done by RNA polymerase. The important words are in red. So RNA polymerase is actually not very important. Why I included it down here is because I'm going to mention that mRNA is translated into protein by ribosome. Ribosome is inside your syllabus, and it is one of the non-membrane organelle that is found in your cell. And the function is to produce protein. And how it produces protein is by translation. So your ribosome will bond single amino acid into a chain of polypeptides. And that's about all you need to know about how DNA codes for protein. Other than that, if you really want to know the difference between DNA and mRNA, you can see that DNA is double-stranded. And as mentioned earlier, the base thymine, T, is replaced by the base uracil, U. And the last significant difference between DNA and RNA is the sugar. The sugar in DNA is deoxyribose sugar. That is where the D comes from, while the sugar in RNA is ribose sugar. The difference between the two sugar is deoxyribose sugar have a hydrogen at the second carbon, while ribose sugar have a OH at the second carbon. Chemically, DNA is less reactive than RNA, if you want to know. But like I said, this is just for your information. What is not just for your information is, let us recall what is alleles. So alleles is different form of the same gene. But what do we mean by different form? Different form just means that the sequence of base is slightly different from each other. This can arise from mutation, and as a result, these might affect the amino acid that are added to the polypeptide chain. This can in turn have an effect on the structure of the whole protein. It can cause the protein to lose its function. For example, when it is halfway through, if there's a mutation which asks the ribosome to stop adding amino acid, the protein form will be truncated. Truncated means shortened. So this will definitely cause the protein to lose its function. But of course, it can have positive effect because, like I said, some mutations are beneficial. So it can cause, let's say, the enzyme to be more effective. That means speed up the rate of the reaction. Or it can even result in a new function of the protein. So this will hopefully bridge the gap that you might have thinking, two alleles, different form of the same gene. What's the meaning of form? So this is the meaning of form. That means sequence of base is slightly different. And that will be all for this lesson.